Welcome back, friends. Welcome to another video tutorial from Shramu's Biology. In this video lecture, we'll be talking about HPLC or high performance liquid chromatography. It's also known as it's a variant of liquid chromatography. We've talked about the gas chromatography, and I told you, like, when you are reading any kind of chromatography lectures and videos, you need to understand three important mechanisms about that. First is what are the mobile and stationary phase in that chromatography. Because these are the two phases that will interact with each other, and the interaction between mobile and stationary phase will ultimately allow to separate compounds from each other. Second is what are the characteristics based on which the separation is conducted. And third is the mechanism by which the separation is conducted. So if you know these three things, you know the idea about the chromatography. In HPLC or high performance liquid chromatography, this is a type of liquid chromatography. So the carrier that actually brings all those sample molecules with each other or inside it is known as liquid. This is the liquid phase of molecule that act as a carrier. In case of gas chromatography, the carrier is gas, for example helium, hydrogen or nitrogen. But in liquid chromatography, the carrier would be liquid. And mobile phase represents to mostly the phase that the carrier presenting. So the mobile phase for HPLC is also liquid, because liquid is carrying all the dissolved substances that we want to separate from each other. So in this case, one thing very important that is required for the chemical compounds that we want to separate should dissolve in that liquid. Now if we talk about the dissolve of the different chemical factors, there are two important things that we need to think about. One is the organic solvent and the aqueous solvent. Aqueous solvent are those which are hydrophilic, where all those aqueous and inorganic molecules solubilized. On the other hand, in the organic solvent, all the other organic molecules generally survive, like they are mostly tend to like hydrophobic in nature. So these two things are very important in perspective of HPLC. So the second question is what characteristics that we use to separate? The answer is solubility, and among that polarity of that compound. Polarity means it could be either polar or it could be hydrophobic. These two characters are the judgmental characters that helps them to separate the chemical compounds using the interaction between mobile and stationary phase. Now the second thing is what is the stationary phase? Whether it's liquid or gas, what? The stationary phase that we use in case of HPLC, it could be either solid or liquid. So it could be solid. Example of some solid stationary phase in HPLC is silica, and it also could be liquid. Okay. Now the third thing is the mechanism with which we separate, that we are going to see in a moment later. The mechanism mostly will be based on separating the polar molecules from the non-polar molecules. Polar molecules from the hydrophobic molecules. Once we separate that, we can easily separate the chemical compounds from each other. Now let's look at the instrumentation process a little bit, and then we'll go to the exact detail of the separation mechanism that is conducted in the column. So this is the complete picture, where it starts with the mobile phase and ultimately ends in the detector, while the detector is connected to a computer, and while the computer has attached to the monitor, and computer uses the data from the detector and put it in the graph, that's going to give us an idea about what is separating and not. Now it starts with this mobile phase, and the mobile phase in this case is a liquid phase that carries a carrier, right? So normally if I simply modify this was earlier I draw for in case of gas chromatography, so I need to modify it a little bit. In this case the mobile phase is nothing but simply it's a solution. So let me draw a chamber that contains a solution. So this is the solution chamber, a liquid solution present here. Okay, like that. So solution chamber that I draw. Okay, and this is the tube which is connected to the chamber. Okay, this is the carrier that is present. This is the solvent which will ultimately carry all the sample with each other through the tube. Now normally it's placed in a chamber, or the outside chamber. Now normally the molecules, either it could be aqueous solution, so it could be like any organic solvent, whatever it is, it cannot flow directly inside the tube. We need some sort of pump that will derive this solvent from this chamber to the tube. So we need to place a pump here at the middle. So there is a pump. Okay, pump is present at the middle, which will help in this flow, start to flow from this chamber to the tube. And right after the pump, we have a sample chamber where we inject the sample. Now the question is, what kind of sample we can use for HPLC? We mostly use biological samples. In gas chromatography, it is not that good to separate biological samples because we heat the sample and make them volatile, make them vapors. But in case of HPLC, we are not heating them. What we are doing here, we are simply allowing this sample to properly interact to the carrier, dissolve in the carrier, and then carrier will carry that sample through the stationary phase, and ultimately it will reach the detector. So in this case, we can use proteins to separate, lipids to separate, and any other organic and cellular molecules and biological molecules to separate from each other as a sample. So we can take a protein mixture as a sample, and then we inject there in the sample chamber. Now let's say here you can see the three different samples with three different colors, black, red and green, that are mixed here, and it's mixed with the carrier, and now it will interact with the stationary phase. Now here comes the most important point about the HPLC process, is the interaction between the mobile phase and stationary phase. Now there are two types of scenarios that can